So hi guys, this is Nikita. Today we'll be revising Cell the Unit of Life. If you are a NEET 2027 or 2028 aspirant, this lecture will be a very important lecture. This is just the first lecture. The subsequent lectures will be available on my channel. So if you like this lecture, don't forget to like, share and subscribe. So as the chapter name suggests, Cell the Unit of Life. It basically means that cell is the structural and functional unit of life. Now we'll start off with the introduction first as it is the most important part of the chapter. There are many PYQs from this part. The physicochemical approach to study biology is known as reductionist biology. G.N. Ramachandran gave the triple helical structure of collagen. Now, he also performed the analysis of protein conformations through the Ramachandran plot. He was influenced by Linus Pauling and his alpha and beta pleated sheets for the structure of collagen. These all things influenced Ramachandran to give the triple helical structure. These can all come as options, please remember it. Now, what is cell? As I just said, cell is a structural and functional unit of life. Unicellular organisms are capable of independent existence as well as performing all the essential functions of life. So that basically tells us in order to be living, we need to at least have a cell. And that's exactly why viruses are not considered living, because they don't have a cellular structure. Now, Robert Hooke observed the dead cog cells. Cog cells are basically the dead tissues of big girthy trees. Then Anton von Leeuwenhoek finally observed the live cells, and Robert Brown gave the nucleus. Now, the microscope and the electron microscope revealed all the structures that we know today. The ribosomes, Golgi bodies, endoplasmic reticulum, all that you could think of, that was possible because of the discovery of microscope and electron microscope. Moving on to the cell theory. Now there's a small change in the new NCERT in the cell theory. So 3 scientists are given in the NCERT for cell theory: Matthias Leiden, Theodor Schwann and Rudolf Virchow. Matthias Leiden is a German botanist. Theodor Schwann is a German zoologist. In the old NCERT it is given as British zoologist. Don't remember that, focus only on the new NCERT. Now Matthias Leiden, being a botanist, only focused on plants. So he gave the law that all the plants are composed of cells. Now Theodor Schwann was a zoologist and he gave a law for both plants and animals. He basically told us that animal cells have a plasma membrane as a limiting boundary, whereas the plant cells have a cell wall as a limiting boundary. But the cell theory did not answer one question, that from where do cells arise? So to clear this doubt, Rudolf Virchow came into the picture. He basically gave us a law which said omnis cellula e cellula, which basically means that new cells arise from pre-existing cells. Moving on to the overview of cell. Now in your 9th or 10th standard, you would have performed an experiment where you observed onion peel cells and human cheek cells. Did you notice something in that? Basically, onion peel cells are plant cells, so they have cell wall as their limiting boundary, and human cheek cells are animal cells, so they have plasma membrane as a limiting boundary. Now in this overview of cell, we basically have to understand what is present in the prokaryotes, eukaryotes and what is present in both of them. Now let us study the common characteristics first. The cell membrane is present in both prokaryotes and eukaryotes. Cell membrane can be present in prokaryotes and eukaryotes or may be absent, like it depends on the situation. DNA and ribosomes are compulsorily present in all the organisms. Semifluid matrix called the cytoplasm or the cytosol is present in both of them. Now, this cytoplasm is the main arena of cellular activities. It keeps the cell in living state. Now, moving on to the prokaryotic characteristics. No membrane-bound organelles are present. They lack a membrane-bound nucleus and they contain 70s type of ribosomes. We'll study more about the ribosomes later. In eukaryotes, both membrane and non-membrane-bound organelles are present. The non-membrane-bound organelles are ribosomes of both 70s and 80s types, and centrosomes which are present in animal cells only. Now, these ribosomes are not only present in the cytoplasm but also in some organelles like mitochondria, chloroplast and rough endoplasmic reticulum. We'll study more about this in future. Now, again a very important question from the PYQ perspective, the cell sizes. The nerve cells are one of the longest cells. The mycoplasma lasts from 0.1 to 0.3 micrometer. Again, this is a little controversial question because in the diagram it is given as 0.1, whereas in the text it is given as 0.3. However, such questions won't be asked. Now the bacterial cells go from 3 to 5 micrometers. Now the human cells have a diameter. Focus on my words. The diameter of 7 micrometers. If they ask radius, they can ask such tricky questions. The radius would be 3.5 micrometers. Please read the questions perfectly. Now the largest isolated cell is the ostrich egg cell. Now moving on to the next part, the prokaryotic cells. Now, the prokaryotic cells are represented by four types of organisms: the bacteria, PPLO, Mycoplasma, and blue green algae. The prokaryotic cells are very small in size, as we know. The bacteria, they are so small we can't see them with naked eyes, but they multiply really fast. And they are found in various shapes or size, but they are fundamentally the same. The shapes of bacteria, again, very important question: Bacillus, Vibrio, Coccus, and Spirillum. Bacillus is the rod shape, Vibrio is a comma shape, Coccus is the round shape, and Spirillum is a spiral shaped. They do not have a well defined nucleus. They have naked genetic material, which basically means that it is not covered by a membrane. Now, it is packed in some form, but it is not packed with histone proteins. This is also a PYQ, please remember that. They have a single chromosomal circular DNA and a plasmid DNA, which is extra chromosomal. Again, a very important question, that a plasmid is the extra chromosomal DNA. The plasmid DNA confers antibiotic resistance. We will study more about this in the biotechnology. If you are a dropper, you know what I mean. They also help the bacteria in transformation. This again you will study in biotechnology, so let's skip that. The prokaryotes have a special structure called inclusions. They are basically extensions of plasma membrane. Now, one such inclusion given in your textbook is mesosomes. Again, they are enfoldings of plasma membrane. They are present in three types: the vesicles, tubules, and lamellae. They help in DNA formation, distribution to daughter cells, respiration, secretion, and increased surface area. Now, again, when we think about respiration, in humans or even the eukaryotes, the respiration is performed by mitochondria. So, we can call this organelle the mitochondria of prokaryotes. Now, moving on to cell envelope and its modifications. Now, bacteria have a very complex cell envelope, which basically explains why they are found everywhere. They have a chemical complex cell envelope, like I just said, tightly bound three-layer structures. There are three layers found in the bacterial envelope. Together, they are protective in functions. Now, let's study the three layers in detail. The outermost layer is the glycocalyx. This could be present in the form of a capsule or a slime layer. The capsule is thick and tough, whereas the slime layer is present in the form of a loose sheath. Now, moving on to the second structure, which is the cell wall. It provides structural support and prevents the cell from bursting. The stain that we put on the cell while observing the colonies is actually taken by the cell wall itself. On the basis of this, the cell is divided into gram-positive and gram-negative. Now, the innermost structure is the plasma membrane. It is selectively permeable in nature and interacts with the outside world. It is quite similar to that of the eukaryotes. We will study about the eukaryotic plasma membrane further in this lecture. In cyanobacteria, there is another inclusion of the plasma membrane called the chromatophores. They contain certain pigments. Now, let's talk about the motility of a bacteria. Now, the bacteria can be motile or non-motile. If motile, they have a flagella. Their flagella basically consists of three parts: the hook, the basal body, and the filament. The filament is the longest structure and it extends from the cell membrane to the outside world. Now, there are other two structures present in the bacteria, but they are not motile in function: the pili and the fimbriae. They basically help in attachment to the rocks. The pili are elongated tubular structures made up of proteins, whereas the fimbriae are small and bristle-like. Now, the next topic that we are going to study is the ribosome and the inclusion bodies. The prokaryotic ribosomes are from 15 to 20 nanometers. They are again located on the plasma membrane. Now, they are basically of the 70S type. The 70S ribosomes consist of two subunits, the 50S and the 30S. One thing that you would notice is that 50S and 30S should make it as 80S. That's because this is a non-additive quantity. You cannot directly add it. Now, several ribosomes sometimes join to a single mRNA and it is called as a polyribosome or a polysome. These ribosomes translate the mRNA into proteins. This again you will study in your 12th standard. Let's move on to inclusion bodies. They are basically for the storage of food material and they are non-membrane bound. Again, all prokaryotic organelles are non-membrane bound. They are of three types: the phosphate granules, cyanophycein granules, and glycogen granules. Sometimes gas vacuoles are also present in the green sulfur bacteria and the purple sulfur bacteria. Now, moving on to the eukaryotes. They are again represented by four types of cells: the protists, plants, fungi, and animals. They have extensive compartmentalization because of the membrane-bound organelles. The cell is well organized with a nucleus and an envelope. Even though they are all eukaryotes, they are not identical in structure. Let's take an example of plants and animals. The plant cell contains a cell wall, whereas the animal cells don't. And the animal cells contain a centriole, whereas the plant cells do not. Moving on to the most interesting part of this chapter, let's study about the plasma membrane. Now, after the advent of the electron microscope in 1950, we could study more about the plasma membrane. Scientists chose the human RBC to study the plasma membrane. Now, moving on to the structure of plasma membrane. The plasma membrane basically consists of four types of groups: the lipids, cholesterol, carbohydrates, and proteins. The lipids are present in the form of a phospholipid bilayer. The proteins present are of two types. They could be integral proteins or the peripheral proteins. The integral proteins are completely embedded in the plasma membrane. They are harder to extract, whereas the peripheral proteins are present on the surface and are easier to extract. Along with these, the cholesterol and the carbohydrates are also present in the plasma membrane. The cholesterol basically stabilizes the membrane, whereas the carbohydrates are present as extensions of the membrane. Now, if you would have studied the ABO blood grouping, you would remember the presence of a sugar on the membrane of RBC. These are similar to the carbohydrates present on the membrane. Now, let's move on to the actual structure of the plasma membrane. Like I said, it is present in the phospholipid bilayer. Now, the phospholipid basically has two parts: the polar head and a non-polar tail of saturated hydrocarbons. The head part is hydrophilic in nature, that means it's water-loving. Therefore, it is present on the outer side of the membrane. Whereas the tail part is hydrophobic, that means it does not like any water. Thus, it is present on the inner side of the membrane, as you can see in this diagram. The integral protein, like I said, is present in the middle of the membrane. It is completely embedded in the membrane. Whereas the peripheral proteins are located on the outer side of the membrane along with the sugar. Now, a better model of the plasma membrane was explained by Singer and Nicholson in 1972. It was called the fluid mosaic model. It basically explained the quasi-fluid nature of the plasma membrane, which is the semi-fluid nature, and how it allows lateral movement. This is measured in terms of fluidity. Now, moving on to the final topic of today, transport of molecules. As we all know, the plasma membrane is selectively permeable in nature. If any molecules wants to go inside the cell, it can basically do so by four methods. The first method is the passive transport. It occurs without the expenditure of energy. Many molecules in our body perform this passive transport. The other method to do so is by diffusion. Diffusion basically means transport along the concentration gradient. Many neutral solutes perform this method as well. Now the third method is osmosis. Osmosis is basically also transport along the concentration gradient. However, osmosis is performed by water only. The fourth and the last method is the active transport. It is basically against the concentration gradient. The active transport basically requires a carrier or a transmembrane protein. Example for the active transport is the sodium and the potassium pump. So that was it for today, guys. More lectures will be available soon on the same channel, so don't forget to like, share, subscribe and drop down a comment. I'll see you guys in the next video. Till then, bye bye and study well.